Vets had been doing it for like centuries before we started doing it in people. They were like, you guys didn't do this?
Yeah, if you guys are interested in like the science of like microbes inside of you, Brad and I are actually going to be on a panel tomorrow about, it's called You've Got a Friend in You. So we're going to talk a lot about that. I don't actually know if we have like poop transplant stuff yet. We'll add that.
I like thinking about the philosophical implications of stuff like this. And you know, if you ever take like Philosophy 101, they always have a discussion, what is it to be you? If I lose an arm, am I still me? Am I someone else, right? So when you think about, you know, bacteria outnumber your cells 10 to 1, and their genes outnumber your genes 1,000 to 1, like, what does it mean to be you? If you think about microorganisms generating oxygen required for us to evolve to where we are now, where would we be without microorganisms? We wouldn't be alive like we are now, even in this room, right? We need the microorganisms that are associated with our bodies to eat, to digest stuff, right? To defend ourselves against pathogens even. So I don't know if anyone has any musings about that. Like, I don't know, does that profoundly change or in any way change the way that you perceive yourself or the value of microorganisms?
Yeah. What does it mean to be with someone else's poop inside? We have more, okay, yes.
I've actually been reading a lot of research on the use of fecal transplants and cultivating the proper, I guess, environment for the right bacteria to grow in there, and it having to do with mental health. And that, when you talk about that, that kind of screams that to me of, okay, so there are all these thousands of microbes living in our body that could, if they're in the wrong balance, you know, making us sad, which is kind of crazy to me.
It is. That's still an area of huge open research right now.
Yeah. So in the lab I used to be associated with, we do a lot of stuff with bioreactors. And so we have taken the human body and we look at it as a bioreactor. That's kind of our unique spin on it. And so we look at this as an inlet and an outlet, and then we have like a hydraulic retention time or something, which is how long it takes something to pass through. And when we're looking at something like an anaerobic digester or something at a wastewater treatment plant that we would use to ferment a bunch of waste, we can look at our bodies in a similar way. Like our gut is a big anaerobic digester, and there are certain microbes in there that will turn certain compounds into certain other compounds. And it's still very preliminary, but some of the thought is now, if you have microorganisms that generate compounds that are, say, precursors to certain neurotransmitters, right? The microbes in your gut might make you feel really happy or really sad. It's literally like you are what you eat, right? So the microorganisms might impact your mental health, which, I don't know, mind blown.
All right.
There's a man in the back.
Oh, okay. Sir with the black shirt. That's you. That's you.
So first, I want to say that I had a really long philosophical discussion with my boss about the value of poop. She lectured me on sanitation protocols.
Who's your boss?
So we use an autoclave. Every day. Are there other bacteria that I should be worried about surviving in the autoclave?
You know, I would not worry about like things from thermal vents in the bottom of the ocean, how to get there. You know what I'm saying? Like, I went in my deep sea submarine and brought it back, contaminated this.
Do you work in
quitting
research
now.
lab?
I work at an animal shelter, actually.
Oh, okay.
I was about to say, tattoo parlor, should I not go there?
You might be okay in an animal shelter. I know that like the microorganism I use, for example, that can survive an autoclave. So it's not going to like infect anyone, but if you are concerned with looking at, say, which microorganisms are present in a certain sample, you have to worry about contamination in that case. That'll throw off your results. Otherwise, I think there's nothing really to worry about.
Yeah, unless you work in an animal shelter where you have like Cthulhu, I think you'll be okay.
Are there any squid in your...
White. Oh no, checkered shirt back there. You had a question way back. All right.
So you were talking about how bacteria converts like inputs to certain whatever type of output you want. What's like the turnaround time usually?
Well, actually, there's a lot of optimization that occurs with like trying to produce outputs from microbes. In fact, most of the field of bioengineering in general is like you have this great idea, you can prove it works, but that's like a 4% efficiency rate. And so a lot of research goes into like actually getting fast turnaround times and high yield. I feel like Brad and Andy actually probably know a lot about that, and Cassandra too.
Yeah, I mean, at least in my experiences, I started out doing a lot of like bacterial work, and so something just like making a protein, let's say I had a bacteria and I decided to throw in some genes to make it red, something super simple like that, come back the next day, red too, you know? And then I started doing stuff with mammalian cells, and I'm sitting here like 3 days later, you know? And then these plant people over here are just like, ha! So it depends on what you're doing. And grad students, we waste a lot of invest a lot of our time in making things do what we want within a certain time frame.
Yeah, I mean, you gotta remember, like when you are doing plant biotech research, like there are ways to speed up production, but they're still plants. They take freaking forever to grow. You have to water them every day. They have to have like excellent nutritional balance, the optimal amount of sunlight.
Gotta sing to them.
You gotta sing to them. You gotta cuddle with them. You know, we take our vaccine production really seriously. If you're not cuddling with your plants, you're doing it wrong.
That's why I'm a big fan of bacteria as opposed to
Yes.
plants. So bacteria tend to grow a lot faster. But I can't remember who mentioned efficiency.
Yeah.
So I think efficiency is always a fun question because people always ask me, well, how efficient is it? It's like, does that really, I mean, it matters, does it really matter? Like if I could paint 100% of this room, but it took me 100 years, would you hire me? Probably not, right? So a lot of times, even in these biofields and stuff, we're looking at kinetics. So if we're 10% efficient, but we could do it really fast, or in the case of the microbial field stuff I used to do, like my microorganisms like high temperatures, so they're really, really, really fast, and they also happen to be efficient too. And so sometimes, you know, it's the idea of faster is almost better than efficiency at times, right? So I would say that's one of the biggest barriers in a lot of these technologies is how do you do it very, very quickly at very, very large scale, right? So yeah, good question.
White tank top here.
So you were talking about the microbes that were like hundreds of thousands of years old. How do they determine that? Like, what do they use to figure out the age?
Yeah, they basically radiometric dating. I'm not an expert at it by any means, though. So, but they essentially date the ice that it was found in.
Okay.
But I don't know if anybody is an expert on this.
It's like carbon isotopes.
It's not carbon.
Yeah,
oh no, you're right.
Andy's got the right kind of like train of thought. So like another really big area of like bioengineering is using carbon dating and various like mineral datings. There's other kinds of dating than carbon that have like longer half-lives. And so because the radioactive conditions at the time were different when those microbes were existing, that's an issue with it.
Yeah, it may be that they there's some certain like signature in the cells that is like characteristic of the atmosphere was different 100,000 years ago. So if you see that signature, here's your clues. But this is a huge field that is really, really well established, and it's very complicated too.
We just don't know about it.
Yeah, we're not experts in that.
Okay.
Yes.
You had mentioned about splicing and cutting up DNA and recombinants and such. Where's too far? Who regulates?
This is a huge question.
I just want to say before you get into the question, I don't do any of that stuff.
I love finding. You're a liar. You have to edit those microbes and get to do what you want.
Yeah. There are millions of microorganisms out there that can do all kinds of different stuff. And for me, it's a matter of I put them in a reactor that will basically select for the ones that do what it is I want. So I use evolutionary principles, and I get the result I want. So
I bet I can modify them to make them better.
Yeah. Other side of the table right here.
2 billion years of evolution. 2 billion years of evolution. It's hard to beat.
Yeah, actually.
I mean, we still got planes to fly though. Birds fly. I'm just gonna say, like, I'm pro engineering in this end. We can do a lot of really amazing stuff. But that's going to be a personal question for a lot of people, which is difficult because we have to make policy choices, right? So as far as, plants and things, there's some things just make sense. We want stuff to be drought resistant. We want to feed people things with greater nutrition. Editing ourselves is a big question that we'll get to in the next 5 to 10 years.
Yeah.
So start thinking, everyone.
There's like one other bacteria that I was considering talking about instead of agrobacteria and chemopassions. It's called S. pyogenes, and it is the organism that we got CRISPR-Cas9 from. I'm sure you guys have all heard about CRISPR-Cas9 in the news. And basically the reason that I didn't want to talk about CRISPR is because I can get up here and talk about modifying plants to make oranges into blueberries and, you know, make potatoes into vaccines all I want. But if I were to get up here and say, oh, here's this bacteria, we can use it to make people have like 4 heads and 6 eyes, like, it's not a good thing. We don't want to get the ethical debate here, you know? That's a little bit too much.
It is Comic-Con.
This is a very open question that I think, yeah, a lot of people are going to have very complicated answers to. Like, just to throw out my brief spiel on it, and again, this could be an entire panel in and of itself.
I wish it was this year, honestly.
Yeah, I wish it did too.
ASU does a lot of it.
Yeah, so I mean, humans have modified our environment to more or less extent since we've existed. Like if you look at crops, the things that we eat now are nothing like the crops that we ate 1,000 years ago. So to me, there's no difference between like, okay, the corn that we're eating, we've genetically selected it by breeding. It's like, look at dogs, okay, dogs used to be wolves, now they're not. So in that sense, I don't have a problem with genetic modification if we're doing it to, you know, make more nutritious food, make us have less diseases, stuff like that. Now, if you're talking about letting somebody see, like in X-rays or something like that, or you know, it gets crazier, then
yeah. I should nuance what I said a little bit. I'm definitely not anti-GMO. As a matter of fact, this discussion reminded me. So the cyanobacteria that I showed here are actually genetically modified. That also goes on in my lab, and
I knew it.
But it also comes back to 2 billion years of evolution is hard to beat. So you can put whatever you want into these microorganisms, but it's really, really hard for them to maintain that because you put them in a situation where they're doing what you want, which isn't necessarily what's best for them. And so they'll lose whatever you put in, or they'll just be outcompeted by any natural thing that comes in to beat it. So that's another thing about GMOs is, yeah, there's a lot of, you know, unknowns about what might happen if they go out of control, but there's also a lot of restraints on what we can even do with them because it's so hard to make a GMO that can win sometimes.
Yeah, for those of you guys familiar with Jurassic Park, there's this lysine deficiency that they put into like the dinosaurs so that they escape, you know. Yeah, it is silly. It's very silly. Yeah, exactly. But you know, you can do certain things if people are worried. If it's like a human or something that they're making, or like a biopharmaceutical that they're making.
No one's making humans right now.
I don't know what these guys are doing. Wait, there's
making plants more nutritious. Slow down.
I'm not just talking about it.
You can basically modify like the genes that you put into the plants to have them have like deficiencies or like other things like that. And so we are doing modification with something dangerous. It is kind of commonplace to put like a vegetable switch into it, like have something that we make it like super allergic to, I don't know, like sucrose. I don't know.
Or it can't live without you feeding it a certain thing, that kind of stuff. So
yeah, we don't want our creations to get out of the lab. We want to sell them. You know,
but as we all learned from Jurassic Park, things can go wrong anyway. So obviously we need to, you know, take things with a grain of salt.
Yeah.
All right, please. Yes, you in the back.
What percentage of the genes or the bacteria or whatever are they naturally selected versus genetically modified?
Oh, I mean, at this point, people were really not that smart. So it's like we're making collages from things that nature has already made. So we're going into some art museum and cutting out the pretty pictures we like and putting them together. We are not smart enough to go and write, you know, a gene that says, you know, be red even. Like we are simply stealing things and shuffling around within life. We are not to the point yet where we can write certain functions, which is part of the reason that something like CRISPR-Cas9, discovering that ability in a bacteria to go in and edit and cut, or these crazy superpowers that we're talking about today, it's so valuable to study those, because those are the tools that we then go cut and paste elsewhere and use for ourselves. So maybe in the future, we'll have people that are really synthetically making genes, but right now, even in a genetically modified crop, right, it's like one gene that comes from a different species that we're usually putting in. So it's just taking one thing from somewhere else in life, putting it into another thing.
Yeah, back to the whole point of like efficiency to like outcomes. like, it would be kind of cool to just make like a giant collage of a strawberry that has like the flavors of every single fruit. But the practicality of actually like, you know, changing nature in a way that doesn't actually help it very much, because you know, when you think about it, these things have been alive for like 2 billion years.
Yeah.
They're pretty optimized already. And so there isn't really much of a need to do much more than insert 1 or 2 genes into any given product.
It'd be like a $10,000 strawberry by Versace or something. Yeah.
This is a big one. Would you fund it?
Yeah. Okay.
Earlier you guys have mentioned like putting bacteria on the surface of like the space shuttle just to see how it would, you know, be. Is there any like research that has been done that shows any sort of like mutations or any advancements that the bacteria do themselves like in space due to like the new environment?
Oh yeah, they do all kinds of crazy stuff. So there's some pseudomonads, which are one of my favorite genuses of bacteria because they just do everything. Nerd. Yeah. Please. They will eat caffeine just as their like sole food source, and they were culturing some of these in space. They didn't even modify them, nothing, and they grow in these like 3D structures with little towers and like channels, and just make this like maze in the microgravity. So